Assalamualaikum and greetings to all. Welcome to the first module of Certified Training Program on Immunization for Pharmacists, CTPIP. I'm Hafzan Hanafia, and I will be your instructor on the Module 1 on Introduction to Vaccines. In this module, we will be focusing on the history of vaccine development, how we can relate the history and current practice in vaccine development and findings. Later, we will be able to relate the importance of vaccination in our healthcare system that leads to immunization and control of the vaccine-preventable disease. There are a few terms used in describing the history of vaccine development: inoculation, variolation, vaccination, and immunization. Inoculation is the practice of intentionally exposing someone to matter from smallpox pustules in order to initiate a mild protective response to the disease. Smallpox inoculation or insertion is the term derived from the horticulture procedures, when a bud was inserted into a plant. Thus, the practice of cutaneous inoculation of material from smallpox pustules later came to be called variolation. Variolation is when there is a use of a lancet or a needle to introduce pulverized, reduced to fine particles dried smallpox scabs or pustule fluid into the skin of an individual. The next two terms is always used interchangeably, although it may differ in the definition. Vaccination is an act of introducing a vaccine into the body to produce immunity to a specific disease, whereas immunization is a process by which a person becomes protected against a disease through vaccination. Hence, not necessarily that a person who is vaccinated will be immunized. When we mention about vaccine development, it involves the process of taking a new antigen or immunogen that is identified in the research progress and developing this substance into a final vaccine. This vaccine shall continue to be evaluated through preclinical and clinical studies to determine the safety and efficacy of the resultant vaccine. Where does variolation or inoculation comes from? Variolation comes from the word variola, which is a scientific name for the smallpox virus. Variolation was a primitive technique of immunizing people against smallpox. People were intentionally infected with a mild case of smallpox to prevent a more serious deadly case. Smallpox is contagious and often spreads through inhalation. After an incubation period of 12 to 50 days, the lesions will appear on the mucous membrane, followed by appearing on the skin. Recorded history has suggested there are two possible origins: China or India. In Asia, variolation technique was developed by practitioners using the alchemical principles as early as the 15th century, although another version reported that it was invented by a Buddhist monk. On the 10th century, it was reported that a nun was practicing insufflation. The first attempt was done with smallpox. The smallpox scabs were pulverized and implanted by blowing the prepared matter into the nostril using a silver tube. The person then contracted a mild form of smallpox disease when there will be a slight fever, which is on the following day increases greatly. In 2 or 3 days, more eruption appears, charged with matters and scab. When they finally recovered, the person developed an immunity to smallpox, although a very small portion of 1 to 2% of those variolated died. This is a good comparison for those who actually died from the infection without immunization by 30%. Some of the recipes found pertaining to this production is really tedious, such as the priming to body temperature condition over a period of time or expose the scabs to hot steam and herbs. This condition may damage the smallpox virus and reduce the infectious load. This is a little different from natural infection. Although similar particles reported of the dry scabs being grinded to powder, the powdered scabs were also mixed with water at times. Another approach done at that time was also by collecting fluid from a pustule onto a cotton flag and placed up the nose. Let us look to another Asian country, India. Claims of inoculation practice in India is described in ancient Sanskrit texts. In 18th century, the English residents in India give descriptions of inoculations done by Indian Brahmin using a sharp iron needle that is dipped into a smallpox pustule, and then the skin is punctured repeatedly in small circles, usually on the upper arm. On the other hand, there are some marked similarity of the methods used in India and in many parts of the Ottoman Empire. By 1700, the variolation technique had spread to Africa, India, and the Ottoman Empire. In contrast to Asian and Africans who inoculated by blowing dried smallpox scabs up to the nose, they tended to inoculate through a puncture in the skin. Inoculation had become established in the Ottoman Empire. Thanks to Lady Mary Wortley Montagu, the wife of the British ambassador to Turkey, who had a first encounter of the variolation practice while in Constantinople. Despite the success of getting immunized using the variolation technique, there are still cases of variolated individuals spreading the smallpox to other people. In 1796, a British physician, Edward Jenner, had encountered another method of inoculation. Based on his observations on a local milkmaid who did not have any oozing smallpox lesions, although they are contracted with cowpox. Jenner used matter from cowpox pustules to inoculate patients, starting off with a young boy who is successfully developed the immunity against smallpox, which is caused by a related virus. New and effective vaccine development must proceed with a detailed understanding of the immunologic responses and protection. It is crucial for designing the vaccine as well as the parameters when evaluating vaccine efficacy. Despite such advancement in technologies, there is still a slow progression in new vaccine development. If we investigate the vaccines currently available before COVID-19 vaccines were introduced, they were developed by determining the components that consistently stimulated antibody responses in infected patients. Most of the vaccines were formulated together with aluminum hydroxide as the adjuvant. Strengthening immunization by WHO expanded program on immunization. Based on the emerging success of the smallpox program, the World Health Organization (WHO) had launched the expanded program on immunization in 1974. The initial goals were to ensure that every child received protection against 6 childhood diseases, which is tuberculosis, polio, diphtheria, pertussis, tetanus, and measles, by the time they were 1 year of age, and to give tetanus toxoid vaccinations to women to protect them and their newborns against tetanus. By 1990, more than 80% of the world's children were protected from the 6 main EPIs during vaccination. Many countries are consistently updating and adding new vaccines to the EPI programs. Since the new vaccine had become available in Malaysia too, the smallpox vaccines was the first to be introduced in the early 1950s. It was followed by the triple antigen diphtheria, pertussis, and tetanus, which continues today. Tetanus toxoid vaccination for pregnant women began in 1974, whereas BCG was introduced in 1961. Polio vaccination was introduced in 1972 due to the polio epidemic, and we have seen such a decline in cases as Malaysia was polio-free from 1985 onwards. However, there has been a resurgence of polio in 1992, with 2 cases among unimmunized children who had history of contact with individuals that has polio. No further cases were seen after that, and the country was declared polio-free by WHO. In 1982, as measles immunization was introduced, rubella vaccination for girls was introduced in 1985. What are the issues and barriers in vaccine development that most researchers are facing now? Generally, there is inadequate clinical data and lack of detailed information on protective correlates of immunity contribute to product failure in clinical trials. Some of the vaccines that is still newly introduced have lack of information on the infectious exposure of the intended vaccine recipients, thus leads to public concern. Vaccines are generally not advisable to be used in population with less responsive. So how shall we overcome this? There is a need to utilize a more relevant animal modules as well as human samples collection. By increasing the use of experimental human, it may challenge the infections. The researchers need to have an open access to information or sharing of databases and findings, as a greater understanding of the mechanism of action may allow faster delivery system. What are our expectations toward the safety of vaccines in the NIP? Will I have side effects from the vaccine? How do I know that the vaccinations are safe? Can I trust the person who makes and gives me the vaccines? Since I have heard that there has been a lot of side effects occurred due to the vaccines, will I experience them? Vaccines used in NIPs are safe and effective. However, like other pharmaceutical products, vaccines are not completely risk-free. Adverse events will occasionally result from vaccination. The general public has low tolerance to any adverse events following vaccination because vaccines are given to healthy persons to prevent diseases. For this reason, a higher standard of safety is expected of immunizations compared with medication that are used to treat people. Each year, vaccine prevents more than 2.5 million child deaths globally, an additional 2 million child deaths could be prevented each year through immunization with currently available vaccines. Why are vaccines so special? Vaccine promotes health unlike many other health interventions. They help healthy people to stay healthy, removing a major obstacle to human development. Vaccines have an expansive reach. They protect individuals, communities, and entire population by the eradication of the infectious disease through a deliberate effort. No further control measures are required, such as smallpox. Another one is that vaccines have rapid impact. The impact of most vaccines on communities and population is almost immediate. For example, between 2000 and 2008, vaccination reduced global death from measles. And then vaccines save life and costs. Recently, a panel of distinguished economists put expanded immunization coverage for children in 4th place on a list of 30 cost-effective management of welfare development. Why is it important to receive immunization? Vaccine-preventable disease or VPD are an ever-present threat. They affect all ages, children, adult, and elderly, and can result in life-threatening complications or even death. Many VPDs are highly infectious and can spread very rapidly throughout entire communities. Thanks to advancement in medicine, immunization can protect us from many once life-threatening diseases such as tuberculosis, polio, and pneumococcal disease. If you are still not convinced, the following facts offer more evidence of why you should immunize. Immunization protects us from the unseen. Vaccine-preventable diseases are not completely gone. Malaysia may be polio-free, but polio still devastates its community in Afghanistan, Nigeria, Pakistan, and Philippines. In this day and age, where we can travel across the world in a day, it is not uncommon for disease to do the same. People love to enjoy food and drink and traveling. Everyone loves to eat, especially while doing their traveling. The food at a stall or restaurant may have been undercooked or prepared in an unhygienic manner, or being contaminated by a handler. This may put us at a risk of infectious VPD that are spread through food and drinks, such as hepatitis A, cholera, and typhoid. Visitors may need to check where that they are going to go. Thus, there has a lot of countries that is actually required you to have a restriction on entry. Visitors may need to check the type of vaccination to be taken before entry, since you may contract a VPD from infected fellow passengers, the lockers of foods and drinks. The risk is increased upon traveling to countries where certain infectious VPDs are present. Immunization has rid the world of smallpox and made Malaysia polio-free. Smallpox is an untreatable disease that once killed 1 in every 3 infected people. And polio is a frightening disease that paralyzes or kills millions of children around the world. Immunization is the only defense against certain diseases. Diseases such as chronic hepatitis B and polio remains incurable despite medical advances. Immunization protects us from cancers. There are vaccines available that can prevent cancers. For example, the human papillomavirus and the hepatitis B vaccines that is against cervical cancer and liver cancer. Immunization cannot be replaced by clean water and better hygiene. Diseases like chickenpox spread from person to person despite clean water and better hygiene, and only vaccines can stop this disease. Immunization can protect the entire communities. When enough of us are vaccinated against the disease, there will be too few left to carry and spread it to others. Thus, immunization doesn't just protect us as individuals, it saves lives. It protects our entire family, community, and country too. This is called as herd immunity. Immunization has ensured healthy life. With new vaccines, we are now able to protect ourselves against previously unpreventable disease, and thus immunization has saved millions of lives, and also it saves money. Why do I say it saves money? Because one of the things that we need to put into account is that when we are sick, we will not be able to go to work, and also the expenditure of medication would be much higher to cater for the symptoms that need to be treated. We have reached to the end of the section. In summary, we had the opportunity to look into the history and current practice in vaccine development and findings, and finally be able to discuss the importance of vaccination that lead to immunization and control of the vaccine-preventable disease. This is not the end. There are more sections waiting to be explored. We shall now have a look into vaccines and its mechanism, and I will see you in the next section.